Good morning, everybody. It's my true pleasure to welcome and introduce our second public speaker, Amy Bernard from the Allen Institute. I met Amy in March 2019 at the Allen Institute, and I have much learned from her since then. Amy has over 30 years of experience and expertise in bioscience and technology at academic and nonprofit research organizations. She joined the Allen Institute in 2005. From 2009 to 2016, she led the research and development and structured science teams at the Allen Institute for Brain Science, and she oversaw the generation of petabytes of data for large-scale open science initiatives such as the Allen Connectivity Atlas, the Allen Developing Brain Atlas, and the Allen Human Brain Atlas, which is outside of HuBMAP focus, but I think we all will want to see a brain ultimately in the human healthy adult reference. Amy then served as a product architect and led the technology product program to deliver open data and tools to the research community through the brain-map.org portal. And I hope you all had a chance to look at that portal. I think it's a very good example of how to get it right. Since 2019, Amy serves as Director for Science and Technology Strategy, providing strategic guidance for the Allen Institute's research portfolio. Amy is extremely active on advisory boards for the NIH Kidney Precision Medicine Project, HuBMAP, and also the SPARK program, both of which are extremely relevant, again, for our HuBMAP work. Today, Amy will present to us on cell type nomenclature. Amy, please.
Thank you, Kati. It's a pleasure to be here. I'm just going to now switch to share my screen. Great. So good morning or good afternoon, everybody, wherever you may be. Again, it's very much my pleasure to be here. I'd like to thank the organizers. I'm excited to be present here for your HuBMAP Year 3 session on day 2. Yesterday, it sounds like there was a lot of really interesting presentations and discussion going on, and I hope that continues today. So today, I'm going to tell you a little bit about work that the Allen Institute has been doing recently on trying to establish a common cell type nomenclature system. So perhaps some of you know more about the Allen Institute in terms of standards related to anatomical structures, specifically brain maps for the mouse, for the human, and also the non-human primate. We've also been working for the last really almost 7 years now on developing the richer model of where those cells fit into those anatomical models. And I just wanted to start out with this quote because I feel that it really anchors a lot of what we do when we think about organizing information, organizing natural structures, anatomical structures. I feel that Stephen Jay Gould really captured this, that in so many cases there are not necessarily natural definitions. Certainly anatomical definitions usually have some anchoring in a functional feature. And for that reason, how we name things or how we organize them is usually very much biased in our own human biases for how we like to think about things. And we rely on our senses, whether it's our visual senses or other senses, to think about how best to partition out information. And nothing says that more, that when we start talking about anatomical structures and organizing those structures, where we're looking at scientific salient features, what are the best ways of presenting that, where we capture the biology, but are still able to set up a system by which we understand and can analyze that information. So here on the left, I actually am showing a little wheel that is derived from some of the work that we've been doing that's published by Tasic et al. In both 2018 and 2020 publications, where we present some of the work that is looking at individual cell types derived from, in this case, the mouse brain, the adult mouse brain. And we've presented this in a way that it's visually appealing, but it tells you a little bit, if you go into the details of the figure, about the number of cells we profiled, the classification of cells were profiled by transcriptomic analysis of looking at mRNA expression, and then how those different classifiers relate to each other in terms of their nearest neighbors and the similarities or differences in terms of co-clustering for those features within those cells. We've done an awful lot of work over the last few years, in addition to others, as part of the Brain Initiative, focused specifically on first the mouse brain, but also the human brain. And to that end, we have been generating just vast quantities of data. There are a couple of reviews here that just show you examples of where this has been published, not just by us, but others, where parcellation of information from anatomical structures is broken down into cells and cell level information that's then presented in these ubiquitous T-SNE plots or HuBMAP plots, or mapped along into a taxonomy of parts. And yesterday we heard a little bit from Alexander talk about the complex ontologies that underlie these taxonomies. And this is wonderful. This makes for beautiful figures. It makes us feel very happy that we can learn so much about the brain in terms of being able to show a representation of gene expression brain-wide. However, we find that when there are so many different collections of information now coming from multiple investigators, we're stuck with very, very complex and massive collections of data that are disparate and oftentimes come with disparate definitions for how that data was collected. So, even though we have the ability to analyze the data, we can look at salient features of the data, we come back into a space where we have sort of improperly posed language to even share and communicate what that data is when we're thinking about the fundamental modalities reflected in cell types. And this becomes important too, as we're looking to coordinate across multiple different groups, different teams to validate the data, as well as to learn from the data that is coming from this large quantity of information. So how do we do that? How do we compare cells across labs? And how do we compare those data sets in ways that are going to be most meaningful for us to extract out the biological features that we're interested in understanding, and then also to provide some sort of terminology or convention by which we can actually then really dig into and share and analyze the data collectively. And for this, I have a picture here of a Rosetta Stone. We always come back to the need of standards pretty much in any kind of science. There's a need to set some sort of a definition, to come up with feature sets that capture the reality of what you're looking at, that allow you to really engage in the most important part, but then also to communicate that information, formalize that information, and then share that information. And so to that end, we sought to find a common cell type or cell taxonomy nomenclature system. Now, there are other nomenclature systems certainly out there for cells, but one of the challenges that we have here with the Brain Initiative is that we're looking for specific modalities that do not right now have any other precedent. So within the features specifically of transcriptomic data, we're now looking at constellations of gene expression patterns that are the anchor points that we would like to use as a very fundamental feature to help us define and then share our understanding of cell types across the field. So, oops, pardon me. What's in a name? Yesterday we heard a little bit about how the most important part, of course, is an ontology, but we still need names. We need names for things. And there's a rich history in neuroscience, at least, for naming cells. We usually name them by their shape or their most apparent function. If we see one that points far, we'll say it's a pointy neuron or a long neuron, or use more elaborate things like chandelier if it looks like a chandelier. We also sometimes rely on features of neuropeptide transmission or production. So parvalbumin positive cells, for example, generate obviously lots of parvalbumin. But fundamentally, these things can be transient, and they can also be highly variable when evaluated by different labs, different communities. And so sometimes it comes down to something as basic as, once we analyze that data or distill the data, we get into some really sort of arbitrary tag. Here we have a cell that's a red one and a blue one and a purple one. And what are these salient features that would actually allow us to use this information if, for example, the colors were changed? What is telling us a functional piece of information about this cell? And for us, one of the things that due to the technology over the last really 5+ years, one of the key anchors in looking at cell types, not just across the brain, but throughout the entire body, is looking at its transcriptomic state. So of course, there's still other properties of cells that are key for their function, but something that is most accessible and arguably most reliable by the criteria of standards definition is to specifically look at the features of cells as they are derived through single cell transcriptomic data. So the good definitions are of course more important than the good names. And they're also the thing in which people present first. So anytime you go to a paper and you're looking at the information that's in there, you don't necessarily pay as much attention to the name, but you're going to pay attention to the information that gathered that name. And what we're seeing is this real preponderance now of many different laboratories generating information about single cell data across all these different initiatives, where they're presenting within usually the publication their own sort of rubric for how those names have come to be. And that may be that they present with a large quantity of data, a taxonomy. It may be that they make some assumptions about the presence or absence of certain marker genes or present proteins or even anatomical structures that are subanatomical structures of the cell, that they're then able to look at and use that within the context of their own study to say, aha, this is actually the anchor for which we're now thinking about the transcriptomic data, and then we're going to use a classification scheme there. And the challenge with this is that we're now getting a different nomenclature system with each publication or with each laboratory. So each study is essentially giving rise to its own, oftentimes unstated convention for nomenclature. And this is really a challenge as all of these groups are seeking to unify the data. They're looking not just to get the information for their one study, but they're looking to get it in a way that it can now be applied for multiple studies and shared more broadly. And nothing speaks to this more than a project like HuBMAP, which is very specifically, the goal is to connect the data sets and the anatomy across the entire human body. So as we're looking to integrate information from other projects as well, be it from the Human Cell Atlas or the Brain Initiative, Kidney Precision Medicine Project, LungMap, all of these projects are also coming up with their own way of not just generating data for cells, but naming them. So if you consider two practical aspects of nomenclature that come from here, the question is, how should we refer to a cell type? You know, what are the principles by which one would even come about starting to do this? Knowing that historically, there are salient properties that people would use about that cell to identify it. When we're looking at all cells of the body, sometimes where we don't even know what the function is, we need to have something that's a little bit more generalized. And so, you know, for the Allen Institute, we thought about this a lot, and we thought, well, this is something, it shouldn't be just us, it should be about the community. How does the community actually want to name things? But we realized that for us generating large quantities of data, we had to start somewhere. And with our own data, we thought, well, maybe we can use as an example a nomenclature system that we're just starting with. And once we start with that, we can get feedback from the community as to how they like it. This is actually an approach that was very similar to what we did with our mouse brain atlas and our common coordinate framework for anatomy. We started with something that based on practical aspects of what the field had already aligned upon. But then we developed and we built on features and functionalities to help that be more useful for people. And I think that we've been fairly successful in that space. And then there's another very practical aspect to nomenclature, and that's about how do we track and match cell types so that we can analyze across many different data sets or data modalities. So for us, in terms of thinking about nomenclature, the data analysis was a really big driver. So even though it's useful to sometimes use the morphology of a cell, particularly in neuroscience, it's very satisfying to look to the shape of a cell. We find that that is not, at this time at least, a really high throughput approach that is also embraced by all of these other fields, whereas something like single cell transcriptomics through RNA sequencing is. And so the data analysis focused on this particular modality is something where we really chose to start, but hoped to be able to accomplish a schema where we could actually use other modalities as well with the same kind of rubric. So this is aligned with, but not directly under the concept of ontologies. So ontologies are important and we want to fit them in, but this is actually really outside of the scope of thinking about the ontology itself and just focused on what I think of as a somewhat, not entirely mundane issue, but as was mentioned yesterday, not the most important issue until you get into the devil of the details of needing to analyze the data. How do you name those things you wish to analyze? Okay, so to that end, we actually had a community challenge in June 2019 at the Allen Institute, where we brought together people from multiple different projects who were working on technologies related to generating large quantities of data. And through that, we sought feedback from them on how do we define some sort of in this case, we say ontological framework, but we were thinking both in terms at this workshop of ontology and nomenclature for a cell type taxonomy. What are the considerations that we want to bring to bear here? And at this meeting, we had lots of lively discussion and very little agreement on the exact best way of doing it. But what did come through is that we wanted to start with the data, to actually use real data to start bootstrapping an approach that we could gather community feedback behind. So we weren't going to be able to come up with anything that was perfect. We realized this would be an iterative process, but we sought to start somewhere. And so we worked with various teams, certainly with ontologists as well as data scientists, anatomists, to put together some of the key issues that we thought were important in thinking about a nomenclature. And this is just a snapshot. Some of the people who are here might even be watching today. There are a number of people who are involved in HuBMAP as well as other collaborative projects. And I wanted to particularly call out the gentleman here in the red shirt, Jeremy Miller, who's really been behind a very, very large portion for our efforts at the Allen Institute to develop and then come up with the nomenclature system and then the applications that I'm going to walk through a little bit today, just in terms of the how-to component for how you might want to use this yourself in your own work. So coming back to this question of what should we call a cell type. So as I mentioned before, there's no perfect approach. Instead, we wanted to do something that was going to be extensible and applicable to our own data. We wanted something that was going to be able to work with classical names as much as possible. So we didn't want to just overwrite or undermine any existing schemas that were there. When there were canonical marker genes or anatomical structures, for example, we wanted a system that could capture that and then appropriately map it onto the right kind of area of a nomenclature schema. We also wanted to be able to use cross-species and cross-modalities. So something that was too focused on a particular species, that embedded, say, a gene name that was arcane or something that was specific to just one particular application, was not as extensible as we would hope. So many of us use model systems such as mouse in addition to human, and ultimately our goal would be to set up something for which embedded in a nomenclature system would be the inference that we would be able to use this for multiple different species as well. And then we also were interested, as I'd said before, of using features that maybe went beyond transcriptomic data. So for much of what the data that we were generating in terms of the transcriptomic space, that is where we would come up with some sort of a taxonomic reference, but it was not limiting to what we understood were functions or identities of the cells themselves. So we wanted, whenever possible, to be able to use a nomenclature system that could capture something like the location of a cell. If you think of the cerebral cortex in the brain, you can think that we oftentimes use laminar structure as a way of referring to cell type, a layer 5 pyramidal cell, for example, or a projection target, long versus short projecting cells. So we wanted something, at least for the brain, and you can imagine this would be the case for other organ systems as well, that had something that was related to its anatomical bearing, because that is so often a case for which people need to have some sort of connection with past studies. So the caveats of the historical approaches, when we looked at what systems were already out there, because there are systems out there now that do capture nomenclature, is that in some cases, they were only applicable to a selected anatomical structure. So for example, if a nomenclature system was only optimized to lung, that wouldn't necessarily be something that those who are studying brain or kidney would be able to leverage. And so we wanted something that would be extensible to any organ system. It also sometimes, we find, includes assumptions about cell properties. We wanted to remove that whenever possible. So if the definition of a type, say, in some cases, the somatostatin positive cells that we see, if you actually profile a somatostatin positive cell by some kind of assay, such as an antibody or protein detection assay, you may not always see that it is in fact expressing somatostatin. It may express the RNA, but it may not actually express the protein. So a somatostatin positive cell may be something in terms of a cell type that's more transient. We also know that, for example, locating a cell in terms of just its anatomy can also be tricky. For example, when we talk about neurons as brain cells, it's pretty obvious that neurons are specialized structures that are in the brain. But what about the vasculature? So the vasculature is also in the brain. And how do we think about border systems that transcend an anatomical structure? So we would like to have some sort of nomenclature system that is flexible enough that can accommodate that. We also know that in some cases, particularly numerical systems where there's a nomenclature system, it may not actually be sufficient as we learn more about cell types. So we suspect that there are thousands of cell types in the brain, and across the body, who knows how many cell types there are. How do we accommodate now these large taxonomies where we're learning more and more about what is defining those cell types? And ultimately, we really want this to be data-driven. So for this to be the most useful, we want it to be something for which we're removing our own biases, and we're actually looking to the data to define what these nomenclature classes should be. So the proposal that we had for this is essentially just a very, very simple structure that we have taken essentially from the concepts that are presented in any sort of classical framework or thinking of gene structure. So genetic identities that are coming forth, and the nomenclature that relies on ID systems is the simple system that we have here. It's focusing on a class and then a format for which we would have a anatomical component, a gene component, a cell class component that's tied to a taxonomy, and then what we're calling a historical name, or in some cases an alias. So something, at least for the neuroscience example, that could capture all of these, but then is layered into a generalizable schema that is, again, similar to the kind of schema that relies on identifiers and then classifiers. So how do we implement this? What we just decided to do was essentially set up a GitHub repository that allows people to walk through how we actually can take a taxonomy that we've generated from analyzing the data and essentially then pin on nomenclature using this framework. So this is very much a work in progress. Your analysis would need to be done in terms of looking at your own cells that you get from your own model system, and then without changing necessarily any definitions, this would allow you to sort of pin on definitions that are consistent with a nomenclature that we've now established. So this is still in development. It's not fully static. We're actually very interested in getting feedback from the community and have a forum dedicated specifically to this that you can access through the Allen Institute website or through this link here. The way that it would be implemented is essentially you need to have a dendrogram. It doesn't necessarily have to be hierarchical, but it needs to be something for which you have conceptualized your data space, your data universe, if you're thinking about individual cells and how they relate. So that could be in some cases also related to an ontology, but it doesn't have to be. You have to then, for implementation, essentially have some sort of a metadata file that we walk through in the GitHub repository for the cluster assignments of your cells. So if you're thinking about a group of cells and what's similar about them, and you've done that analysis, you need to then make sure that you've annotated which of those cells fit into that cluster. And then you'll need to go through and have some time and expertise to actually annotate those cells. Okay, so what does that look like, at least for the GitHub repository? You just go to our GitHub site and you'll see this little page that describes to you essentially what I've just said, what this is for. The general concept, again, is that you've already collected your cells or data from your cells, and you've defined some form of a taxonomy, or you've found a taxonomy that you like that works well for your particular cell types. You then would define or pin additional cell types based on that cell type organization. We like to use a hierarchical organization, but one of the things we learned at our workshop was that that was too limiting, and it shouldn't be embedded in terms of how one thinks about a nomenclature system. So in some cases, you can think where hierarchies would either be be integrated with other systems. For example, when I was talking about the vascular system with the brain, you wouldn't necessarily have a hierarchical system that was separate and sub-regionalized to brain that was separate from the vasculature of the rest of the body. So while a hierarchy may be good for some organ systems, it's not necessarily something for which it should be a requirement in a nomenclature system. And then the annotation and standard terminology that is assigned to this common cell type nomenclature framework, that's where we actually have essentially just set up a system where it pins on an ID. So there's an ID that would go with that specific cell type, but you can have any particular other alias that goes with it. Again, much as you think about how our gene accession IDs work, you can have a gene ID, and then you'll have sort of a vernacular description that goes with it that accommodates the flexibility for essentially the different stages of analysis that have brought the field to that particular nomenclature term. Okay, so what are the specific parts that we have now in this nomenclature system? So I'm just going to walk through some of the parts that if you go through and you looked through how to actually implement this, these are the terms that apply. So the cell set component is a key concept that just essentially relates to any set of cells that are within a specific taxonomy. So the taxonomy that you could apply, it could be, for example, in brain cell types, that's what we've been using, but it could really be anything. It could be a kidney, could be a sub-anatomical region, or it could be something that is not even related to features that are limited to an organ system. It could be an internal node. It could be a part of a hierarchical system as well, but it doesn't have to be. It could be any cells that were collected from a particular donor, or just cells that are related to a structure, or even any just cells that you think through your own analysis were really cool because they all had some sort of salient feature that you don't fully understand yet. But that's essentially how we think of a cell set, and that's a term that we're now using that's part of the requirement that would get you into applying the nomenclature framework onto a particular a set of cells in the framework. So for our purposes, you can track anything you want, but we're actually now looking at cell types or groups of cell types. So for the work that we've been doing at the Allen Institute on the brain, our cell set that we're using is actually looking at cell types. So another key concept, this came up a little bit yesterday as well, is with the taxonomy. So a taxonomy is just really here defined as a quantitatively derived data cluster through which we've run a specific computational algorithm on that specific data set. And the information about this process would be tagged and given a label. So that this would involve metadata of what is that analysis that was run. So that would be a description of that analysis. And then it would be a description of that corresponding data set that gave rise to that analysis. And then there would be an ID that's given to that. So for example, in this case, we have a taxonomy accession ID that we've created. And I'll talk a little bit more at the end of this presentation about, sort of longer term vision for how to collect these taxonomies, because we don't have an answer for that yet. So the taxonomy accession ID, here's just an example. This is a value that matches what the cell set and this particular process that would have been done has metadata on it. And for here, we're actually using this just in a date format. So we find that for the accession ID, this is just a useful term that we have, but it could be anything. We then have a short name for what the critical information is about the taxonomy. For us, it includes, a reference to our organization, Allen Institute. We're then using transcriptomic, or in this case, T, as a modality that we're analyzing. This is our third version that we're doing, and then we've appended it with a species tag so that we know this particular taxonomy is related to human. I should point out that this could be a shared taxonomy, human and mouse, for example, in which case you could append it with something that captures that. There's then a longer name that includes additional information that is for people that want to start including information, say, that starts digging into a little bit more of the modality that they're using. So here we just say transcriptomic T, but in this longer name, we actually specify that we're using a SmartSeq version 4 as our experimental access to the data, as opposed to, say, Illumina 10x. There are other ways that we could get gene expression data, and we may choose to capture that in the particular taxonomy. For us, then, the subset of this particular taxonomy includes the cortex, which was included. In some cases, this is the mammalian cortex of the brain. We also have generated now taxonomies that are broader, that cover the whole cortex and the whole brain as well for mouse and human. We then look at a confidence threshold for clustering dendrograms. In this case, our taxonomy actually did include a dendrogram, and for us, we have confidence thresholds for that. We then include a date. The species is there as well. So this is, again, the kind of metadata that we would see, and then versioning of this. All this encapsulated within our definition of taxonomy descriptor that is in the GitHub repo. Okay, so another key term that we have here, of course, needs an ID. So the cell set, we talked about the cell set before as being any kind of set of cells that you're looking at. And that accession ID is what we now track across all data sets and taxonomies. So the cell set accession ID, it's linked to the taxonomy. You can see here through there, and hence appended to it as well, so that we understand, and it essentially marries the notion of a particular set of information tied to the taxonomy that together defines what that nomenclature will be. So when we were actually developing this, we got feedback from the community. We'd originally started with just 2-year slots in the ID. People are very ambitious and seem to think we need 4 slots in the ID number for that, which would mean we could potentially take it into the next century. And then there was also a suggestion that we increase the number of cell sets beyond 1,000, which is, again, I would say ambitious, but in a good way. Okay, so that accession ID, of course, is again the equivalent to like an entree ID for people who are into the genomic side of things. And this has just a little bit of sort of embedded information. So it's not a completely random number. It relates back to the taxonomy ID and then also to the cell set ID, but it's also not a fully user-friendly name. And so for that, we also have an alias. And this, although it doesn't seem user-friendly, it is user-friendly for biologists, because what it does is it starts explaining for us what other sort of cell types might be called, say in a paper, in a figure legend, in somebody's vernaculars. They're describing the categories of what those cell descriptors are that they're thinking of cell types. So in this case, it could be an inhibitory layer 1-2 PAX6 positive, CDH positive, ADARB2 positive cell from ganglionic eminence. This actually does make sense to a lot of neuroscientists, and hence it's actually a useful alias that also captures the descriptors that are in that particular cell type. But it's linked then to another term. Okay, so then the other thing that is related to the cell alias is what we think of as an aligned alias. We started calling this as a preferred alias. But we found that that was maybe a little too loaded for some users. And so the idea of an aligned alias is one for which there's hopefully, and this is where there's a little bit of, there's just the grayness that one gets into with nomenclature. The user-friendly term that people commonly understand that links across different sorts of analysis types. So in this case, this would be sort of analogous to when somebody thinks about a gene symbol just as a single word, as I'd said, maybe, parvalbumin. It's something for which people now associate the attributes that a deeper analytical dive gave rise to, but has a more vernacular name. And in this case, this would be a chandelier cell. So chandelier cells had been described prior to any sort of transcriptomic analysis a long time ago by Ramoni, Cajal, and others through their morphological properties. And we now have information through looking at a deep taxonomy of looking at gene expression profiles that now aligns with what this chandelier cell is as well as a descriptor. And so this is a field that might, the term, at least in the field, is what would link it across different potential researchers or papers and potentially data sets through this aligned alias, this alias here. And this actually then requires that people analyze their data, that this is where you fit it into a broader perspective of what are the features that are similar to your studies and other people's studies. Okay, so this is just sort of walking through a little bit of an example of the cell types and then the groups of cell types that we had in a recent publication that would be present in any taxonomy that we're doing using this nomenclature schema. Essentially, you start with a cell set accession ID here. That's what I described before. So that says there's a particular set of cells that were part of this transcriptomic analysis that had been anchored in a given taxonomy that was done and analyzed separately. And for this case, the cell set alias, for us, this is cell type, are biologically based names. This particular example here, as I'd said, it includes inhibitory cells, layer 1, layer 2, here shown in this particular side of the dendrogram. So this is just the terminology that we've been using at the Allen Institute. It's extensible, but it doesn't have to be absolute, and anybody could use that particular definition. So I've just lofted here. What we're now adding on is the accession IDs to the alias, which was the term that we'd used that was based in our example. And shaded in the background, you'll see here there's little sort of blue nodes. What those blue nodes are showing you how we can look at groups of cell types. So groups of cell types are also cell set aliases that you can define. And in this case, for our cell set aliases, we also have IDs now. We have the descriptors, and this is actually now focused on specific nodes. And for those nodes, that's essentially looking at this as a new cell set. So for that cell set, we also have an ID. And so in this way, we can then layer on essentially multiple types of analysis states that anchor back again to how we've analyzed the data, to a given taxonomy for the data, and then to the relationship between those that is now quantifiable in a way where we have a terminology to set it with. And so, why is this important? If you look at some of these nodes, these are nodes that we were interested in getting some additional validation for using multimodal data. So this particular analysis that I'm looking at here was just focused on RNA sequence analysis, looking at gene expression values that had been essentially mapped, and a taxonomy of relationships had been created only using that transcriptomic data. But that's just one potential descriptor of cell type characteristics and properties, and we were interested in seeing to the extent to which that would align across multimodal data as well. So in a recent publication now that is in bioRxiv, we have multimodal data using a patch-seq technique, which is patching on to a neuron, a recording from its electrophysiological properties, and this is just shown in the panel on the left, mapping that spatially to where that cell is located in a common coordinate framework, then removing the nucleus and subjecting that to transcriptomic profiling so that we will identify what the RNA sequences are in sort of nucleus and some of the surrounding cell media. And then backfilling that cell so that we actually get information about its anatomical and morphological properties through just a spatial filling process where we restructure that cell. So using this technique that was really established by the Tolius lab, and we've now been using it en masse to profile thousands and thousands of cells, we wanted to essentially use this multimodal data approach to inform the cell type nomenclature. A, to make sure we were comfortable with our nodes that we were looking at, and B, so that we could start looking at this again as a little bit more of a Rosetta Stone, where we could link other people's data types, whether it be morphological or descriptive, into what is now coming forth for our transcriptomic data set that has many thousands and thousands of cells, actually heading into millions of cells. And so this was just an example of how, again, we're sort of using the Rosetta Stone of multimodal data and then pinning this essentially to the nodes that were shown here in terms of understanding what kind of cell types we're looking at and a little bit more of their physiological properties. So an application of this particular nomenclature framework to the non-transcriptomic data is described, as I'd said, also in this paper here. So this is where we've taken what we're calling the morphoelectric cell types from mouse visual cortex, meaning we have morphology and electrophysiological recordings from these cell types. In this particular example, we did not have the component that was transcriptomic. And then we seek to pin that on to the transcriptomic data. And we see that in this particular example, 11 of the 20 morphoelectric types that we've identified are shown as ones that we can pin on essentially just using information from their morphological and electrophysiological properties to look at cell types. So this is where we essentially use the cell set identifier. We used example physiological data as well as morphological data to then map to a known cell set ID. And this is now, this is one of multiple papers that we've published that we can sit here and now go back, look at the data, and then apply on the nomenclature so that we can compare it into a broader growing data set that we have. Okay, so another example in terms of matching cell types between data sets. We may have interest in looking at data sets that go beyond our own, and we're interested in also being able to do that. So this in theory is extensible. I think that one of the challenge is actually there are multiple different taxonomies where different teams, different laboratories are generating measurements potentially of the same cell types and are coming up with different aliases. So for us, we were also interested in seeing whether or not the clusters derived from, for example, 2 different analysis techniques could be put together into cell types that we could have a shared taxonomy for. So in this particular example, we look at cells from human transcriptomic data, mouse transcriptomic data. As I'd said before, we looked at the electrophysiological and morphological properties and those properties together. And for this, we actually then generated in a number of different publications, multiple different ways of describing that data. So we've now gone back and mapped on sort of to a shared nomenclature what those cell types are. And so this is just in terms of the BRAIN initiative recently. The desire had been to come up with a whole bunch of different cell types that had preferred aliases, that also had descriptors from previous publications as well as our own work. And now what we've essentially done, in addition to the ones that are listed here on the right, which is non-neuronal cells, so cells still that are in the brain, but that we're not able to get either electrophysiological information from, or do not fill into it or feed into a taxonomy that is specifically related to neuronal properties. We're able to then map those again into a format and an example that includes a reference back into the shared nomenclature that we've described, but should be extensible as you map that up to a higher level taxonomy. So an example for a specific cell type, again, coming back to our favorite of the chandelier cells, is the GABAergic cortical interneurons that described in this Bakken et al. Publication that just came back this last year, that we mapped specifically to corresponding data that we'd seen from other cell taxonomies. So this is across both mouse and human brain, where we'd looked specifically at the properties that were in a shared taxonomy that we'd created, and then mapped to the alternate alias as well as a preferred alias, all of which you'll see have slightly different gene expression characteristics when you look at that node. So that's something that is interesting, because what it allows you to do as well is, if you were just looking, for example, at the gene expression properties through the taxonomy assignment that was through just the transcriptomic data, you may or may not have realized that these are also the shared cell type that is described through many, many other publications of the chandelier cell. And you're also able to see that between mouse and humans, so for here under species, we indicate where it's human and where it's mouse, that there's some variability in terms of what those marker genes are. And this is, again, an important consideration in thinking about use of marker genes and transferability across data sets, and then how those can actually inform when you're looking at broader data sets, say, and you want to do cross-species comparisons. This is a way that then would allow us, and in essence the broader community, to understand what the relationship is between that primary analysis of the transcriptomic data, where then again there's modalities that could inform something even more. So for example, some of our morphoelectric modalities were what helped us assign and pin on the chandelier term into this particular cell type. So that's just an example, and I just wanted to have a few more comments related to implementation. So in terms of next step, we have been asked many times about governance, who's governing this, what's the centralized databasing. For our own purposes, we do have now our own at the Allen Institute way of just using this. So we are implementing it through our own use, but we're interested in getting feedback. How you can participate is you can try mapping it yourself. So if you generate cell type data and you're interested in assigning nomenclature, try it and let us know how it works. You're probably already formalizing your cell sets, people do. And so it's really just about assigning an ID to that in a way that would be consistent. In terms of future work for adoption and governance, there's immediate uses, of course, to the schema. We're using it. And we're interested to see whether or not people actually want there to be a centralized storage or repository, or whether or not people don't. So this came up at the workshop that we had. A lot of people wanted there to be some sort of NCBI style of centralized storage, where other people thought that wasn't necessary. So again, this is just about starting to get feedback from the community. We are working on our own type of a service that would be able to ingest, store, and view other people's taxonomies. We're hoping to be able to generate that in 2021, but right now we're only focused on the brain. So how this would be useful for other communities remains to be seen. And in principle, this could be implemented for others, but we wouldn't at the Allen Institute be able to host that. So that's where it just could be a really good community approach to seeing how, not so much a technological solution, but just a very practical way of sharing data could come to be. This is common issue throughout the field. So we have not addressed governance, and this was again a question we thought for nomenclature. What is the best way of doing that? Is it best to have a single entity that manages it? Is it a community-based framework that is self-organizing? We don't know. We suppose that some sort of supplemental work that I think is just now getting funded by the NIH may be able to help inform this, but ultimately that's something that has not yet been defined. And so really we're interested in getting feedback from people. Again, as I'd said, we published right now papers under review in eLife. There's an archive preprint here on the description of this in more detail. It's also on our website. Every slide has shown this particular URL, or if you go to the alleninstitute.org and the brain-map.org website, you can see a full description, essentially walking through what the justification is and then pointing to the GitHub repository as well. We also wanted to just quickly call out that Peter Karchenko's group is for Human Cell Atlas is working on a sort of cell annotation platform, which is a much broader integrated platform for integrating components of not just identifying the cells, but then also applying a nomenclature. And this would be consistent with that. So with that, I just wanted to quickly close with the aspiration that having reference taxonomies would of course be desirable. Right now, everybody can roll their own taxonomy, but there's not a centralized space for cell type taxonomies. And we think this is important. We would love to see NCBI establish cell type taxonomy in there. They already have taxonomies, but they're specific to species, not cell types. And we think that that would be a really good sort of future space for this to go, so that there's a more formalized definition around the taxonomies themselves. So it's not perfect, it's a start. It's going to be iterative. The framework for the taxonomies themselves is not yet here, but we believe that still people are developing and very much in the throes of developing their own cell type taxonomies, and we're excited to see where that goes. So with that, I just wanted to thank you all for your attention, and we have time for questions. And I wanted to particularly thank Jeremy Miller, who again is the brains behind most of what's gone on into certainly our GitHub repository, and also just in terms of all of the Allen Institute scientists who've been working on the cell types data, specifically data related to the NIH Brain Initiative that's part of the BICCN and BCDC network, which is still very much of a work in progress. And I know Kadi's always asking me, when are we going to have all the cell types in brain? And I keep telling her that we're working on it. So I'm going to stop sharing my screen, I think, and open it up for questions.
Thank you so very much, Amy, for your great talk. We do have, in fact, a few questions here, and we're going to start with the following one. I'm going to copy and paste this into the chat here so that everybody can see it. And the question is, have you found cell types that only exist within the mouse brain and not in the human brain?
That is a great question. So I would say no, but we have not yet exhausted the cell types of either. So we've been very comprehensive about our search for cell types in the cortex. So in the mouse and the human cortex specifically, we have not found cell types as such that are just specific to mouse that we don't suspect there's some form of homologue with in human. But the reverse is not the case. So we have found a couple of cells in human where we have not yet had convincing evidence that there are corresponding ones in mouse. Or, and this is actually where their nomenclature comes into play, it's not clear at which node in the taxonomic tree those cells are mapped to. So if you go high enough up the nodes in the tree, it may be they are homologous, and the specialization is where we're a little bit thrown off. But I would say, you know, accurately, the jury is still out until we actually have mapped all the cells. We can't say for sure. And that's, again, just looking at transcriptomic profiling, not looking at multimodal cells.
Thanks very much, Amy. Seems like that was quite a question to open up with.
It's loaded, yes.
Yeah, so the next question is a little bit longer. I put it into the chat box and I'm going to read it real quick. So could the nomenclature alignment benefit from the creation of a semantic ontological map of the space? It seems like there could be ways to show and decompose the nomenclature into, when visualized, may help people come to a consensus. This process is also data-driven and can be updated.
Yes, so yes, I'm so glad that question came up. It's actually interesting because when we had our workshop, the premise for what the best way to do cell nomenclature was exactly that. So not just a semantic, but also a fully data-driven approach to using all data, integrating it, and then using that output as a way of developing the nodes. And the challenge actually we found when we started sort of unpacking and getting to what would it take to get there was still the diversity that we had from all different labs in getting to a framework where that was possible. And so it's actually really interesting because this is where formalization of taxonomy paired with cell set becomes so key. That to get to the point where you would be able to do that, you still have to pair some form of a taxonomy with some form of a defined cell set. And once you have that, then yes, I think the ideal way to do it would absolutely be to be taking more of a machine learning approach to it, or a semantic approach to gather the data in a way where it would really be sort of a whole picture together. But that's, again, sort of the staging challenge that we have. I think of the field right now, there you can think the phases of projects are forming, or storming, forming, norming. And at least in terms of the space of cell diversity, certainly in the brain and thinking more broadly for HuBMAP across the whole body, we do not yet have a complete picture of what all these cells are as we're getting transcriptomic defined data for them. So it's not until we actually have that raw data that I think that that's going to be a really, really successful and viable solution. And I would love to think that this is where I found it sort of appealingly optimistic when people thought we needed year digits that were 4 digits instead of 2 digits. I don't see the kind of nomenclature terminology that we're working with now as something that should bring us beyond the next 10 or 15 years, because at that point, the hope is that we would really be able to use more of that kind of approach specifically to classify and inform what the relevant taxonomic groups are, as well as the components of those groups for a nomenclature that's actually rooted in the data. So I don't know if that fully answered the question, but it's an excellent point. And I think that what I talked about today is not in any way intended to replace that approach for data clarification.
Thank you so much, Amy. I do have some more questions here. The Q&A section has been quite busy. I'm going to go ahead and post this next question. For anatomical ontologies, this person has seen variations of atlases from 60 parts up to 400 to 500 parts. So are there challenges in embedding cell information specific to parts of the anatomic ontology, given the varying atlases that are out there and that people use?
Yes, of course, absolutely. So I think that's actually one of the greatest challenges, and I've had discussions with probably many people, some of whom are on here now. Of course, it's a challenge, and I think that's where these multimodal approaches really are I think the most successful way of thinking about the solution space. So some of the features that are within these cells, even when we ascribe on a consistent nomenclature to them, they're going to transcend organ systems. And again, I use the vasculature as an example system. So we know that it doesn't in any way take away from their validity within that organ system, but it does mean that we have another modality that we have to attend to in terms of thinking about function. And in terms of the atlases specifically that have so many different parts, the cell level information in terms of embedding it is just, it's simply not possible to do it in completeness. So it's this curation challenge. It's having layers of accessibility to the data where you can either facet to view the layer that you specifically find to be most salient for that study. We've tried to do that with some success, but not complete success, through Allen Institute for Brain Science web products, specifically looking at the anatomical and gene expression framework through visual information of sections. But now that we have entire genomes worth of data in individual cells, it's becoming unwieldy. And then that's when we move to things like, dimensionality reduction, like HuBMAP, to look at a different dimensionality of cells, for example, that maybe you could visually sort of blow out from a reference map that is also spatially anchored. So it starts turning into a little bit of a trick to scale the level at which you want to look at for your cell features. And that just has to be curated largely with a lot of feedback from the user community. So I don't see any sort of magic bullet there. But yes, absolutely, there are challenges. And I think that many of the people here certainly involved in HuBMAP are the ones working on those solutions.
Thanks so much, Amy. Moving on to the next question. This one is a lot shorter. Just posted it here. How is the 3D context of a cell or a cell type captured in the nomenclature?
Oh, that's a great question. And unfortunately, it's not. So you could add that on. You would need to, and in fact, there's a team at the Allen Institute involving Nuno da Costa, Clay Reed, and Forrest Coleman, who are working right now on EM reconstruction of exactly the same space that we've generated an awful lot of our transcriptomic data from. And they're specifically now looking at the synaptic properties at the EM level to define some of those spatial cell types with its own taxonomy. So much as I showed you for the example of the multimodal taxonomy with the morphoelectric data, that's actually now what we're hoping to loft in is some of the very high resolution EM data that would give you that information that's derived for the taxonomic purposes of that cell shape. And so you can imagine then through that, you could come up with essentially the alias that contains information relating to that shape. But it's at this time it's not embedded as a feature. And that was actually a very sort of hot topic of discussion when we'd had a conversation looking at some of the classic cell type definitions that are morphological and spatial, how important that is. So yes, of course it's important. The challenge is then mapping it to a modality for which is comparable with other organ systems. So the reality is that the morphology alone is not going to allow it to be a modality that is usable through the whole entire body, because morphology has, say, less of a feature if you're looking at a liver cell. Well, it still has importance, but it doesn't have the same level of identity. So that's where we came back to the transcriptomics anchor for this particular data set. But it just needs its own taxonomy. And that was one of the things that I was trying to make clear, is that anything that you can create a taxonomy for, in this case, a spatial features taxonomy for which you have enough of a data set to define that, you can generate a nomenclature that works for it using this schema. And then you can map it using the node framework for those cell sets with whatever data you have.
Thank you so much, Amy. All right. I think Kadi wants to have a few more words before we wrap up this part of the morning.
Amy, we wanted to thank you, and all invited speakers get a little thank you trophy. This is the visible human spleen in a miniature version printed in 3D with a scaffold to hold it in place. And thank you so very, very much for coming in, and I will find a way to send this to you via old-fashioned postal mail. Thank you so much.
Thank you.
Yeah, thank you so much, Amy. All right, everybody, we are all thanking Amy Bernard for her fantastic talk. And we are making our way back to the home base. You will find the link in the agenda. And then from there on, we're going to continue our day. All right, see you over there in the home base.